right into chapter 9. We have an activity that I'd like to take the last, hopefully 15 minutes of class and spend some time on. And so let's see what we're doing. Here, we briefly talked about this as you were leaving. Some of you packed up your notes early. This is a big deal when you're talking about irradiating a sample with gamma rays. Can I destroy cells? Yes, and I could make cancerous cells or I could destroy them. You are really talking about exciting this double bond, and it actually allows the electrons here to form covalent bonds with another nucleotide below, which forms these kinks inside of the DNA, and that has the high probability it can lead to cancers, okay? It can also just mess up transcription factors and the way it works and other things, right? And sometimes it's not in coding regions, so it's not at the end of the world, but other times it becomes a big deal. We're talking about probability. 3.8 billion base pairs. They're coming in sets of codons in sets of 3. I got 21,000 genes that we're coding for in my DNA, but a lot of what we still generally call junk DNA that's over there, just where did that mutation happen in a deamidation, a removing of my, what am I thinking of it, depurination, or also my X-rays hitting it, or my UV rays. Okay, other sources for nucleotides, definitely, right? So we mentioned this at the beginning, NAD, FAD, right? Coenzyme A, all part of metabolism that you'll deal with as you move forward, but nucleotides have other purposes, and we looked at some of those as well. Cyclic, secondary messengers, right? Sanger sequencing. Now, we didn't get to talk about Sanger sequencing, right? Nope. Okay, so Sanger sequencing, just so that you all know, is dead. It's gone the way of the dinosaur birth. It's not what we use anymore, okay? But we teach it so that you can get up to speed with what modern techniques are, okay? So in Sanger sequencing, the secret is something where we're going to look at something with a DDN, okay? A double deoxy. So if I'm looking at this, what is this that's making it double deoxy? It's not just DNA now, which had the deoxy on the 2'. I'm also missing it on the 3'. Why would that be a big deal if I were using that to try to use a polymerase and extend a chain? Yeah.
The 3' oxygen has to act as a nucleophile for the addition of the next subunit.
Right. It's where I'm adding the next base pair, the next nucleotide. It's going to go right there. And if it's missing, it cannot extend the chain. It's literally poison. So they actually add about 1% of each of the nucleotides in the double deoxy in Sanger sequencing. And so as it's going around, it's got 99% probability it's going to find the right base pair, put it in, and it's good. But it's also got a 1% odds it's going to mess it up. So with that, I can get varying lengths where sometimes on the very first go, it's already the double deoxy, and all I get is 1. Or I get 1, 2, or 1, 2, 3. But many of them get much longer. So that is why when I'm using this, if I've got a double deoxy and I've got a coding strand that I'm making it onto, and then I run into the double deoxy at these different positions, I've essentially added to a set point, and then this double deoxy hit there, and that's why this is only 1 in length. This was 2 in length. This was 3 in length. And so the length of the actual polymerized nucleotide strain of my matching is short. Now, this is an old way we also did it, where we used radioactive isotopes of it so that we could actually visualize it on gels. We also used other modes that we can see that in. But that, you're looking at sequencing. As you look at that, it's written 5' to 3', because how was it writing? The first one that went off the template strand was, the coding strand was a 3' end, and it was writing 5' to 3'. So it added just the G, then added a G and an A, and then it had been poisoned. So this is how it's read. And so you should be able to easily decode this. Yeah, and this is what's called the sequence. Now, please pay attention to these vocabulary terms. Sequencing, which we're talking about next-gen sequencing, Sanger sequencing, is where I determine the actual letters for my nucleotide polymer, okay? I know what the sequence of those nucleotides is. When we talk about PCR later, PCR is not sequencing. PCR is merely making a new copy of the strand.
Yeah.
It doesn't give me the sequence. It just makes a copy of it, okay? The other thing that I want you to know is this. When I write, please pay attention because you're gonna wanna get it right on the exam. When I write, I'm always writing 5' to 3'. Have I said that before?
Yes.
That is how we write it. What if I had written this as I'm going down and I have the coding strand such that I have my code backwards and I have it written out 3' to 5'? How would the multiple choice naturally write it?
5' to 3'.
It would write it 5' to 3'. So if you're transcribing something and you're going backwards in the way you transcribed it, realize you've got to flip it in order to get it right on the multiple choice exam. Why? Because it will be written what? 5' to 3'. Pay attention to that. It will make sense when you run into it, and if you're not looking, you'll miss it. Did you figure out some way to mark that in your notes? Okay, I know some of you have photographic numbers, it just works great. There are other ways we can do it. Now these are fluorophores. GE contracted with a company and bought these Bodipi background things, and we use them in proteins. We also use them in DNA sequencing. These little fluorophores here were what we were actually doing competition with when I was in graduate work, where we were designing fluorophores as well that could be imaged, and it would come out in different colors when I excited them with lasers, and they would fluoresce in these different colors.
Cool.
So in designing these, there are several different modes that we've designed. Some of them are designed such that I can now excite them, and then the fluorophore will fall off. So I don't need a double deoxy, but I'm blocking the ability to extend it because I actually have positions on that it won't extend if I don't see the fluorescence. Others have other modes to it. So this is what's called next-gen sequencing. Now I'm going to let you watch this clip for just a minute. This is explaining next-gen sequencing faster than I will if I try to talk about it, because I'll give you way too much detail, okay?
Okay.
So this is part of the world that I came from. This is what we're talking about. This is the most popular pattern, one that we have out there now.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a bond, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands
Now
are
this is
firmly
covalently
amplified
attached,
through
right?
bridge amplification. In this
To that
process,
long.
the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters.
Just have all of the
resulting in
nucleotides I need,
amplification
and then
of
the
all the
polymerase,
fragments.
heat, cold, we
After
can
bridge
amplify it.
amplification, the
Cover the
reverse
whole
strands
lot.
are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With
So
each
as
cycle,
it gets in
fluorescent
there,
intact
when it binds, it
nucleotides
actually
compete for addition to the growing
flashes.
chain.
It's literally flashing the
Only
fluorescence
one
as
is
it binds.
incorporated based on the sequence of the template. After the addition of each nucleotide,
Way easier
the
than a double
clusters are
deoxy, you
excited by
have to
a light
separate out,
source,
you just
and a
look for the flash.
characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines.
If you were looking at one spot, remember it's covalently bound, if that's not moving on your lawn, and you're looking at one spot, it's shining a light, and each time you add at that spot, you're literally seeing the new color, and it's flashing, you know it's added the new one, okay? So that spot, you know the sequence. The computer can read all the spots
At the same time.
as they're happening.
And you would just like cycle through the different types of nucleotides? So you just like wash it with one
You know
type,
what colors
wash it
and
with the
which
next
ones,
one?
So that's
It's
what's
the base
happening.
call.
Yep.
For a given
Got it.
cluster, all identical strands
So
are
that
read
was forward
simultaneously.
strand.
Hundreds of millions of clusters are sequenced in a massively parallel process.
This is what it
This
looks
image
like.
represents a small fraction of the flow cell.
Good luck interpreting that.
After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template.
By the way, when you look at those data files, and you're looking at them for the thousands of people that we do, they're petabyte drives that we put them on. Now you guys probably know what a terabyte drive is, right? From megabyte, gigabyte, and then we went to terabyte, petabytes the next 1,000 up. Okay, and you don't send those over the internet, you mail the drives. Okay, and so that's what we were dealing with in the industry all the time. But once you sequenced it and you got that raw data back into just letters, you can have thousands of them as just letters, and it's really very simple to read.
Yeah.
The read is generated similar to the first read. After completion of the index read, the read product is washed off,
So do
and
it
the 3'
forward
ends of the
and then
template are
reverse.
deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized, and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments.
They were short
Sequences
strands
from
because we
whole
cut the
sample
DNA
libraries
up,
are
so
separated
they're
based
short.
on the unique indices introduced
50
during the
to 80
sample
long.
preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward
Align
and
them so
reverse
that the
reads are
sequence
paired,
match.
creating contiguous sequences.
Forward and
These
reverse
contiguous
strands match.
sequences are aligned back to the reference genome for variant
This is the
identification.
original one, the reference genome we
The
did.
paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in BaseSpace Sequence Hub. Discover the possibilities of next gen.
Illumina's, I don't care about them, but they are the biggest leader in that field, okay? Beijing Institute of China has over 1,000 of those that are running, about really 100 of them every day, just running, cranking through that. Yeah.
How long does it take to reroute one of those panels?
Yeah, so it's not as long as you might think. We usually take a day, a little bit less than that, to actually do a sequencing now. Now the goal, when we actually originally sequenced, and I'll show you some of that data as we get into this next unit, was we wanted to get it under $1,000. It's now there, and in less than 24 hours, and it's now there. So we have achieved the goal. Okay. Yeah.
So, do we just, is this all made in a machine, or do you run the solutions individually?
So the solutions are there. Essentially, you buy them pre-made and you just dump them into the machine.
Okay.
So technicians can run this without having to put somebody that's a qualified chemist. But you might guess that the original synthesis and all ideas were all chemistry-based, biochemistry world, right? But and between organic chemistry and biochemistry understanding, this is what we did. But it's done. This work's done. But there are people who are working on ways to make them longer reads, which actually have more accuracy. You can get hundreds of base pairs at a time, and there are some platforms that can do that. But this is the one that's popular because it's cheap and it's really easy. Okay, so overview of transcription. We're going to come back to this in about another 5 minutes because we're still in chapter 8, but this is where we want to be. Okay. So, what do I have at the top?
The parent.
The parent DNA. What's happening?
It's unraveled.
It's unraveled, and then I'm writing what way?
5' to 3'.
I'm writing 5' to 3'. So polymerase is writing 5' to 3'. That's the way it goes. The template strand is in that place, 3', 5', but that's how I'm writing. And then the transcript is created in what? What is this language? This is now, I'm not making a copy. This is RNA. Okay, so RNA, instead of using A to T, goes A to U. Okay, that's fine. And then I use these codons to come down here, and that matches my amino acids. Okay, so that's what we're doing. There is something known as a start codon. Okay, so it's a methionine, and that's where we start. There's always a promoter region upstream that helps us recognize that, but that's where you start the actual read. And then when it gets over, it takes this over to make that. So the RNA, when it's read, how is it read from the standpoint of turning into amino acids? The ribosome reads it 5' to 3'. So that's what's going on, okay? All right. So there's your start and stop codons. There's several stop codons. It is not, it's important that you find the first start codon because you'll shift where my codons are based on where was the first start codon. And so you can't find the stop codon at random. You have to find the stop codon where? Based on where did I start it. Yeah, Nate?
Don't some bacteria have like multiple start codons or like sequences overlapping where you have to start in a row, but it can start any of them and form active genes?
Yeah, so there are some tricky elements there that can happen. Yeah. Okay. So that was where we at. Let me introduce chapter 9, and then we're going to really hop into where we're going. So chapter 9. My goal is to give you 15 minutes. I don't know if I'll make it, but I'll get really close. Okay. So in the meantime, while we're doing this, as we're about to do this activity, let me pass these down the row. This is what we're going to work on for the last bit of class. We're going to actually work on sequencing. Okay. Something you should be able to do already. So many of you are familiar with C.S. Lewis's work. Here in Corinthians we read, therefore, if any man be in Christ, he is a new creature. Old things are passed away. Behold, all things are become new. In Messiah, and thus they became new creatures. Unless they do this, they can in no wise inherit the kingdom of God. I promise you that just like when Eustace in the Narnia series, and C.S. Lewis is writing about him, because he's become a dragon, in order to become the new creature, the one transformed by Aslan here, meaning the Savior, in order for him to become what he needed to do, was it painful? Yes. It was a painful process, and it involved who? The Savior. And does he change us? The answer is yes. Coming to the Savior to let him change you comes at a price. You have to be willing to let the old die. When your agency is willing to let that happen, the Lord can take that piece of you until eventually we become all new, and he can remove it. I promise you and testify, he can do that. Trust him. He knows more than we do. Okay. So in talking about DNA and cloning, becoming new creatures, yes, here's a Harvard professor. This is legit. This is recent research. If I were doing it, back in 21, in September, we're excited. This Harvard professor is given money to get the woolly mammoth brought back from the dead, right? I don't know if that's the right thing or not, but we're excited about it, right? GMOs are a topic of interest I could go on for a while about. Simply said, in the last couple of years, it's become obvious to me that most of you no longer have in your concern the thing that previous generations did, which was a major concern that GMOs were somehow bad, okay? The government invested largely in GMOs. Much of your food crop comes from the fact that it's GMOs. It's existed for more than your generation. And the fear is because we didn't ever mark them as they went out to the field. You didn't know if you were eating a GMO product or not. But what were we doing? And the answer was, we were trying to make it so that these food crops, we were taking a gene from one other plant or from some bacteria, and we were putting it inside of new plants. And so Monsanto is one of the largest ones that's involved with them. I've done business with them. They're a great company to go work for. There are stories of corruption if you go back. Be careful where your sources are. That's all I can say. Okay? But yeah, there's a lot of different options out there that have existed, okay? And there's a lot of false claims. So I got some links in there. Here's some genetically modified things. Sugar beets, Roundup steak, the ability to spray Roundup, which is previously, people used to actually, I know people who used to try to sell Roundup, and there were some salesmen in their staff, they didn't, who actually thought Roundup was so safe they would drink it. It's a glycophosphate. Take a cup of it and drink it, because they really believed it wasn't ever going to harm people. That's a stupid maneuver, okay, to believe. But they thought they could sell more that way. Roundup is, by the way, in probably all of our bodies right now. It's been proven that even if you're in a non-agricultural region, you're in the middle of some industrial and you've never been out there, that you probably are full of Roundup, because it really gets into our food crops. And so, is it a problem? Yeah, it is. But the ability to spray that spray on and not have any weeds, that's really cool. And if used correctly, probably not such a big deal. Incorrectly, problem. Pest resistant, so they don't even have to use a spray. Oh, that's cool. Antibiotic from getting things like the potato blight. That's cool. Okay, so we have a whole bunch of things that are out there. Okay. One here, Simplot, locally in the state, also heavily involved in this. I could have potentially taken a job. When I was graduating, there was a guy who was just hired. They were hiring 20. This last year, I just went down. In fact, this year I just went down. Met with the guy that's managing all of Simplot at a conference, and we talked about their most recent research got going on. with potatoes. They're making them really small. They've removed acrylamides inside of them, so when I say acrylamides, they don't have acrylamide. What amino acid do they have? Arginine. And arginine, when cooked at high temperatures, becomes an acrylamide. But when it polymerizes, not so big a deal. But they worked on removing those. So fantastic, cool chemistry, Maillard reactions, and things like that. We'll talk about DNA cloning when we get back. In the meantime, for the next 12, 13 minutes, your goal is to do something that I find fascinating. Many of you have taken a genetics class or are taking a genetics class. Many of you have taken a biology class where you're convinced you can sequence. You can just write this code down and you can transcribe it right into amino acids. And what I have discovered, what Brother Rubish has discovered, what we've known for the last decade, is that most of you learned it wrong. In fact, when I say most of you, really over half of you, if I had not given any instruction, would have done it wrong. Let's see if you can get it right today. Okay. We already gave you some basic instructions. Let's see if you can get it. You'll know you got it right. You really will, because you'll have it sitting there at Wednesday.
Well, first you'll transcribe all of it.
So there's that, right? So be thinking about what you're writing, and this may be of some help to have a start and start codon up there. I don't know if it's big enough for everybody, but you have the information on your sheet too. Okay?